Good morning, students. So we will have topics on molecular biology today. So looking at these images, what will come to your mind? So this is BT cotton, this is golden rice, and this is BT brinjal, this is dolly, this is CC or copycat. So looking at all these images, so what we get in mind is the molecular biology technique, that is recombinant DNA technology. So these are all the genetically modified crops or animals which has revolutionized the molecular biology as such. So with this background, let us start today's topic on recombinant DNA technology. So before going into the details of this rDNA technology, let us see what are the set objectives for today's session. As already mentioned, rDNA technology has revolutionized molecular biology concept and it is having an increased impact on clinical medicine. So it can be at the level of diagnosis, or it can be at the level of treatment, or at the level of research. So this rDNA technology is popularly known as genetic engineering. Now we will go back to the history of this rDNA technology. So in the year 1953, Watson and Crick, they are the one who described the structure of DNA. Followed by, there were so many discoveries, and in early 1960, Warner Arbor, he is the one who discovered the biological function of restriction endonucleases. Followed by this, in 1967, there was isolation of DNA ligase. So these are the two important enzymes which we will be dealing when we discuss with rDNA technology, that is restriction endonuclease and DNA ligases. Then in 1970, Hamilton Smith isolated restriction endonucleases and it was used by Daniel Nathans in 1971, where he used this enzyme to cut the DNA. Then Peter Loban, he is actually a graduate student of Dale Kaiser. He proposed the idea of rDNA, and it was first developed by Stanley N. Cohen and Herbert W. Boyer. So it is Cohen and Boyer in 1973 who actually developed this rDNA technology or rDNA. And in 1978, the first product of this rDNA, that is the drug which was generated, was human insulin. So it was generated by David Godel and colleagues in 1978, but it was available for marketing during the year 1982, where FDA gave approval for marketing. So this is a brief history of this molecular biology concept, that is structure of DNA, to generating a product, that is protein, by rDNA technology. Now we will familiarize ourselves with few terminologies. So the first one is clone. So what is a clone? It's an identical copy. So what is cloning? It's a production of multiple copies. So it's a technique of producing multiple copies or amplification. So in an easy terminology, we can put it, amplifying the identical copies is nothing but a clone. So they are the genes from same origin. So the cloning can be a cell-based cloning or cell-free cloning, otherwise enzyme-based cloning. So cell-based cloning is nothing but which uses rDNA technology. In cell-free cloning, it uses PCR technique for amplification. So this difference you should note. rDNA technology, it's a cell-based cloning. PCR is a cell-free cloning where you have only the enzyme-based amplification. Now we will move on to this terminology, genetic recombination. So genetic recombination is the exchange of information between two DNA segments. So this recombination actually happens in a same species, and this is one of the common mechanisms which operates in our body to produce many immunoglobulins. So genetic recombination is nothing but exchange of information between two DNA segments. Now coming to genetic engineering, it's an alteration of genetic makeup of an organism using various biotechnology methods. So here in genetic engineering, you are actually altering the genetic makeup. You can use two different species in this genetic engineering. Coming to recombinant DNA technology, now synonymously genetic engineering or recombinant technology are used, but recombinant technology is the technology which involves isolation and manipulation of DNA, including end-to-end joining of DNA from two different sources to form a hybrid DNA. So you have to isolate a DNA of interest, then you have to add it or transfer it onto another DNA fragment where both are joined end to end, and that resultant DNA is known as hybrid DNA or chimeric DNA or recombinant DNA. So this whole process is known as recombinant DNA technology. Now we will see the protocol of rDNA technology. To start with, we have to isolate the DNA. So there is isolation process of DNA. Now this DNA is fragmented by restriction endonucleases where we get a desired gene or desired DNA fragment. Second step is we have to isolate the vector. So whatever vector is required in the process, we have to isolate it, and then this vector is cut open by the same restriction enzyme. Now you have a cut open vector and the desired gene. In the third step, both these fragments are joined end to end by the enzyme ligase, and the resultant product is recombinant vector, which is nothing but a hybrid DNA or chimeric DNA, or otherwise it is called as recombinant DNA. So this hybrid DNA is introduced into the host cell. So it is moved into the host cell. There are so many processes which helps in moving this vector containing the target gene into the host cell. After introducing it into the host cell, there will be amplification, that is the cloning will happen. So there is multiple copies of the same DNA is formed, following which you can use this clones where you can isolate the desired gene, or you can express this gene to produce the desired product. So you have to identify whatever clone you want using so many techniques. You can use hybridization techniques, so on, and then you can express the gene where you can get the desired product. So now you see there are so many methods involved in this technology. So there is isolation method, there is selecting method, there is transforming method. So whatever methods are used, they are collectively referred to as rDNA technology. So the methods to accomplish these and related tasks are collectively referred to as recombinant DNA technology. So it is a combination of many technologies.